Friends, welcome to Pharma Toppers. Myself Piyush Agarwal, and today I'm going to deliver a lecture on Alzheimer disorder, flow of content, introduction, pathways and receptor, protein misfolding, Alzheimer disease, cerebral ischemia. So here you could see in this figure which depicts a difference between the healthy brain and the brain Alzheimer disorder. In this you could see the cerebral cortex, which is responsible for the language and the information processing, whereas in case of Alzheimer disease brain, the cortex the area in the cortex are damaged, which are involved in the thinking, planning and remembering, as well as the ventricles. You could see here the cerebral fluid is grows larger, whereas in case of healthy brain it is within its capacity. And also here you could see there is a normal hippocampus lobe, whereas the hippocampus lobe shrink severely, which is responsible for the memory, or basically the most important nucleus which are involved in this is amygdala nucleus, which converts the short-term memory into the long-term. So here is the difference between the necrosis and the apoptosis. This difference is important, friends, so this must be remembered. The morphological features are mainly important, like loss of membrane integrity in case of necrosis, swelling of cytoplasm and mitochondria, total cell lysis, no vesicle formation and disintegration, whereas in case of apoptosis, membrane blebbing but no loss of integrity, aggregation of chromatin, shrinkage of cytoplasm, fragmentations of the cell into the smaller bodies, formation of apoptotic bodies, and mitochondria becomes leaky due to the pore formation involving the protein of BCL2 family. This point is important. And also physiological significance are to be remembered, that is in case of necrosis affect the contiguous cells. It is evoked by the non-physiological disturbances, phagocytosis by macrophage and significant inflammatory response, whereas in case of apoptosis it affects only the individual cells. It is induced by the physical stimuli, phagocytosis by the macrophage, which is more or less similar, but also by adjacent cells, and there is no inflammatory responses. So apoptosis plays an essential role in embryogenesis, helping to shape organ during the development by the eliminating the cells that have become redundant. T cells have a negative regulatory pathway controlled by the surface programmed cell death receptor, for example PD1 receptor, and that is normally a balance between the stimulatory pathway triggered by antigen and this negative regulatory pathway, apoptosis inducing pathway. So the balance is important in the maintenance of the peripheral tolerance, in that receptor is upregulated on autoreactive T cells. A disturbance in this balance is seen in the autoimmune disorders, in the exhaustion of the T cells, in chronic viral disorders like HIV, and also the tumors which escape from the immune destructions. Apoptosis was found to play a role in monitoring the cancer change because it acts as a free line defense against the mutations, purging cells with abnormal DNA that could be the malignant. Withdrawal of these cell survival factors, which is involved in the continuous signaling, active signaling by the tissue specific trophic factors like cytokines, hormones, cell to cell contact factors like adhesion molecules, integrins, which has been termed as a death by neglect, is not only the pathway to the apoptosis. The death machinery can be activated by the ligands that stimulate the death receptor, that is the death by design, which is important, and by also the DNA damage. Macrophages that are engaged in the clearance of the cell corpse release anti-inflammatory mediators such as TGF beta and IL-10, which are acting as a protective. A protein termed apoptotic initiating factor that is released from the mitochondria enters the nucleus and triggers the cell suicide. So this point is important. Caspase 8 is active in processing the inflammatory cytokines like IL-1 and IL-18. So coming to this figure, friends, as you could see here, the diagram between the two main signaling pathway in the apoptosis. Starting with this, the death receptor pathway is activated when the death receptors, such as members of the tumor necrosis factor family, are stimulated by specific death ligands. So this recruits the adapter protein that activates the caspase 8, so which in turn activate the effector caspase, that is caspase 3. So the mitochondrial pathway is activated by the diverse signal, that is, one being the DNA damage by p53 protein, and in presence of DNA damage that cannot be repaired, activates a sub pathway that results in the cytochrome C from the mitochondria. And from the mitochondria, with subsequent involvement in the apoptosomes and the activation of the initiator caspase, that is caspase 9. This pathway, that is apoptosome and the caspase 9, which is known to be the mitochondrial pathway. So the apoptosome is a complex of the caspase 9, cytochrome C, and the apoptotic activating protease factor type 1 or APAF2. Both this pathway converge on an effector phase caspase, that is caspase 3, which brings about the demise or the death of the cells. The survival factor sub pathway, which is normally holds the apoptosis at bay by inhibiting the mitochondrial pathway through the activation of the anti-apoptotic factor, that is BCL2. So the receptor R labeled represents the respective tropic factors or the growth factors, cell-to-cell contact factors like integrins and all, etc. So continuous stimulations of this receptor is necessary for the cell survival or proliferations. If this pathway is non-functional, so this anti-apoptotic drive is withdrawn, and thus inhibitor of apoptosis, that is been used as a inhibitor for the caspase 3 and thus prevent the apoptosis. So caspase belongs to a family of cystine protease present in the cell in the inactive form. They do not perform generalized proteolysis. They undertake delicate protein surgery, selectively cleaving a specific set of the target proteins like enzymes, structural components, inactivating some and activating others. So a cascade of 9 different caspases take part in bringing about the apoptosis, and some functioning as initiators that transmits the initial apoptotic signal, and some being responsible for the final effector phase of the cell death. So each receptor has a death domain in its cytoplasmic tail, and stimulation of these receptors by the external ligand, that is tumor necrosis factor, as I've explained earlier, or TREL, cause them to get together in the 3, that or trimerize, and thus recruit the adapter protein that complexes with the trimer by associating with the death domains. So the mitochondrial pathway by the DNA damage and by withdrawal of the action of the cell survival factor occurs. The sub pathway involves is P21, the pro-apoptotic branch of the family, that is BAX, which promote the release of cytochrome C, and also the nitric oxide is also found to be the another mediator which have shown the pro-apoptotic and the anti-apoptotic actions. The crosstalk, as I have explained earlier, between the death receptor pathway and the mitochondrial pathway together involves in the apoptosis. So the effector stage of the caspase mainly due to the caspase 3 enzyme and the inactive cell constituents such as the DNA repair enzyme, protein kinase C, and cytoskeletal components. A DNase is activated and cuts genomic DNA between the nucleosomes, generating DNA fragments of approximately 180 base pairs. So here are the examples of the exuberant apoptosis with increase of the cell death includes neurodegenerative disorders, conditions with tissue cell damage or the cell loss like myocardial infarctions, depletion of T cells in the HIV infections, osteoarthritis, hematological disorders like aplastic anemia. Again, example of defective apoptosis include cancer evasions of immune response and resistance to the therapy, autoimmune or the inflammatory disorders, viral infections with ineffective eradication of the virus infected cells. So the BCL2 protein is oncogenic because it inhibits the apoptosis and increase the resistance to the cancer chemotherapy. Other anti-apoptotic members of BCL2 family are BCLXL and MCL type 1, and they are all current targets for the anti-cancer drug. So remember this point, friends. An antisense compound against BCL2, that is oblimersen, are in the phase 3 clinical trials for multiple myeloma and leukemia. So investigations of the antisense compound against MCL1 are in progress. So the death receptor for the ligand such as TRAIL are expressed on the cancer cells and undergo apoptosis when TRAIL binds. So monoclonal antibody to the TRAIL are in the phase 1 for the cancer chemotherapies. A new indirect target is the proteasome for the degradation of the protein, includes those involved in the apoptosis. The drug which are involved is botemosib. A boronate compounds covalently bind to the proteasomes which inhibit the proteasomes for the treatment of selected cancers. So it causes the buildup of BAX, an apoptotic promoter protein of the BCL2 family that acts by inhibiting the anti-apoptotic BCL type 2. So botemosib is recognized as acting partly by inhibiting the NF-kappa B action also. An endogenous caspase inhibitor, that is survivin, occurs in the high concentration in the certain tumors. So compound that inhibits, that is inhibitor of apoptotic protein, leads to the free caspase to induce the cancer cell suicide. So the drug based on the antisense approaches to survivin inhibitions are being considered for the clinical trials. So in this chart you could see, friends, the uptake through the transporters and also the efflux to the transporters of the molecule, and thus the activations of the cytochrome enzymes, the synergistic action with the apoptotic protein and for the apoptosis, and the anti-apoptotic protein which prevents this apoptosis, and together with the antioxidant enzymes. So as a result of which, if this anti-apoptotic agents fails, so the apoptosis continues and thereby inactivations of the DNA enzymes, and thus the there is a apoptosis in the DNA and thus the cell damage. So here you could see, friends, about the protein misfolding here. The native protein which is misfolded and thus forming the oligomers and thereby insoluble aggregates. Thus it is deposited both in intra and extracellular and thus causing the neurotoxicities. The responsible factor here is mutations or the external factors. So chaperone plays a major role in this, which prevent the misfolding of the protein by utilization of the ATP by conversion into the ATP into the ADP2 inorganic phosphate. So misfolding means adoption of the abnormal conformations by certainly normally expressed proteins, such as they tends to form the large insoluble aggregates. The misfolding often means the hydrophobic residues that would normally be buried in the core of the protein are exposed on its surface, when gives the molecule a stronger tendency to aggregate, initially as an oligomer and then as an insoluble microscopic aggregates. They also tend to stick to the cell membranes. So the tendency to adopt such conformations may be favored by specific mutations of the protein or by infections with the prions, which is also a protein. So the production of chaperone protein which bind to newly synthesized protein or the misfolding proteins and encourage them to fold correctly, and the ubiquitination reactions which prevents the protein for the destructions within the cell. So Alzheimer disorder basically was originally defined as a pre-senile dementia, an age-related dementia which was considered to be the result from the steady loss of the neurons that normally goes on throughout the life, possibly accelerated by the failing of blood supply associated with the atherosclerosis. So loss of the cognitive ability with the age is considered to be the normal process whose rate and extent is very variable. Alzheimer disorder is also associated with the brain shrinkage and the localized loss of the neuron, mainly in the hippocampus and the basal forebrain. The loss of the cholinergic neuron in the hippocampus and the frontal cortex is a major feature of this disorder and is thought to underlie the cognitive deficit and also the loss of short-term memory that occurs in Alzheimer's disease. The two main microscopic features are the characteristics of the disorder is amyloid plaques, which is deposited extracellularly, consisting of the amorphous extracellular deposit of the beta amyloid protein or A beta and intraneuronal neurofibrillary tangles comprising filaments of a phosphorylated form of microtubule associated protein, that is tau, both of which these deposits are protein aggregates that results from the misfolding of the native protein. So they appear also in the normal brains but are in smaller numbers. So the symptoms may not develop many years. So the altered processing of amyloid protein from its precursor is now recognized as the key pathogenesis of the Alzheimer disorder. Familial Alzheimer disorder, in which the mutations of the APP gene or amyloid precursor protein gene or of other genes that controls the amyloid processing have been discovered. The APP gene resides on the chromosome 21, which is duplicated in the Down syndrome, in which early AD like dementia occurs in association with the overexpression of the APP. So amyloid deposits consist of the aggregates of A beta containing 40 or 42 residues. A beta 40 is produced normally in small amount, whereas A beta 42 is overproduced as a result of genetic mutations, shows a stronger tendency. A beta 40 and 42 are produced by the proteolytic cleavage of much larger, that is 770 amino acids APP, a membrane protein normally expressed by many cells including the CNS neuron. So the protease that cut out the A beta sequence are known as secretase. So alpha secretase act to release the large extracellular domain as soluble APP. Formation of A beta involves cleavage at two different points, including one in the intramembrane domain of APP by beta or gamma secretase. So gamma secretase was found to be a clumsy enzyme, actually a large intramembrane complex of several proteins that lacks the precision and cuts in APP at the different positions in the transmembrane domain, generating A beta fragments of different lengths, including A beta 40 and 42. So mutations in this region of APP gene affects the preferred cleavage points, tending in the favor of formation of A beta 42. Again, the mutation of unrelated presenilin gene results in the increased activity of the gamma secretase because the presenilin protein is a part of the gamma secretase complex. So these different AD related protein mutations increase the ratio of A beta 42 and 40, which can be detected in the plasma, serving as a marker for familial Alzheimer disorder. Mutations in another gene for lipid transport protein, that is ApoE4, also predisposed to Alzheimer disorder, probably because of the expression of the abnormal ApoE4 proteins that facilitate the aggregation of Alzheimer disorder. So in the other main player of this biochemical stage of Alzheimer disorder is tau, which is already been discussed, a protein of which neurofibrillary tangles are composed, a normal constituent of the neuron being associated with the intracellular microtubules. So in Alzheimer disorder and other tautopathies, it becomes abnormally phosphorylated, enhanced by the presence of A beta plaques, and is deposited intracellularly as a paired helical filaments with a characteristic microscopic features. So when the cell dies, these filaments aggregates as extracellular neurofibrillary tangles. So the tau phosphorylation impairs fast axonal transport, a process that depends on the microtubule stability. So friends, you could see here the structure of amyloid protein precursor. The soluble APP is one of the part of this APP, and this red line indicates the sites of the amyloidogenic mutations, and here are the enzymes, that is alpha beta and gamma secretase, acts on the different sites and thus generating A beta 40 and 42. So again here you could see the processing by two pathways, that is physiological and the amyloidogenic pathway. In physiological, the S-APP is synthesized by growth factor function, which is a question mark, still in questions, and more research is required. And in amyloidogenic pathway, A beta 40 and 42 is being secretes due to the ApoE4 mutations, and also by the action of these secretase enzymes. Thus aggregation took place, and thereby the amyloid plaques cause the neuronal death. And again the phosphorylation occurs by kinase, which is still in the question. How this phosphorylation occurs, or the triggering mechanism of this phosphorylation. So thus paired helical filaments causing the neuronal death. Again it's a question, and research is required, thus forming the neurofibrillary tangles. So dihydroergotamine was used for many years to treat the dementia. It acts as a cerebral vasodilator, but trials showed it to produce little or if any cognitive improvements. Nootropic drugs like piracetam and aniracetam improves the memory if in animal tests, possibly by enhancing the glutamate release, but are probably ineffective in Alzheimer disorder. Memantine, which is an NMD receptor antagonist, are believed to work by inhibiting the glutamate-induced excitotoxicity. So inhibitors of the beta and gamma secretase have been identified and undergoing in the clinical trials. Epidemiological studies reveals that some NSAIDs are used routinely to treat the arthritis, reduce the likelihood of developing the Alzheimer disorder as well. So ibuprofen and indomethacin is mainly used. So it reduced by reducing the A beta 42 formation by regulating the gamma secretase, an effect unrelated to the COX inhibitions by which NSAIDs reduce the inflammation. A beta plaques binds to the copper and zinc, and removal of these metal ions promote the dissolution of the plaques. So the amibicidal drug, that is cliquinol, is a metal chelating agent which is important, that causes the regression of the amyloid deposits in the animal model of Alzheimer disorders and showed some benefit in the initial clinical trials. So cliquinol itself has no toxic effects in the humans, which preclude its routine clinical use, but less toxic metal chelating agents are under investigations. So the shortage of growth factor, mainly the nerve growth factors, may contribute to the loss of the forebrain cholinergic neuron in Alzheimer disorder. So here you could see, friends, the chart in which the current Alzheimer disorder drugs are available, which is used as a treatment, that is tacrine, donepezil, rivastigmine and galantamine. So remember its mechanism of action and the main side effects, and obviously the notes like tacrine, which is found to be effective in Alzheimer disorder, but hepatotoxicity is the most important side effect. And again, in case of galantamine, the dual mechanism of action is postulated. So this chart represents the ischemia, which in which here you could see increase in the RIP2 factor of gene cause the activation of the caspase 1, and thereby this protein is activated, and thus acting on the mitochondria, thus releasing the apoptotic inducing factors and the SMAC gene, and as a result, and as well as the cytochrome C enzyme, and thus forming the apoptosomes, and thus activate caspase 9 as, and again caspase 3, as a result of which cell death occurs. It also activates the caspase 8, so which is responsible also for the death receptor pathway, and here this pathway is the mitochondrial pathway. And these are my references. Thank you for listening to the lectures. For more videos and materials or any queries, feel free to subscribe at www.pharmatoppers.com.